we are going to get started. So we're on Chapter 11, How Genes Are Controlled. For a lot of you that took Bio134, this should actually be review. So how and why genes are regulated. So every somatic cell in a given organism contains identical genetic instructions. They all share the same genome. So what makes cells different from one another? So every single cell in our body in the nucleus has the exact same set of DNA. And our DNA genome has like 3 billion base pairs in it. And so why are our cells so different? Heart, blood, brain, lung, bone. They're all very, very different, perform very different functions, have very different structures, but they all have the same exact information. The process is called cellular differentiation, making cells different. Cells become specialized in both structure and function. And certain genes are turned on and off in the process of gene regulation. So we actually call them gene switches. And when certain genes are turned on, the cell develops certain features, certain structure to perform certain functions. The rest of the genes are turned off. Patterns of gene expression in differentiated cells. So the central dogma of biology states information flows from DNA to RNA to proteins. We talked about that last week during all the processes. We have 3 billion base pairs in our genome, and not all of them are turned on, quote unquote, at the same time. The great differences among cells in an organism must result from the selective expression of genes. So not all 3 billion base pairs are on at the same time. That would be chaos. Bacteria are simple organisms that are easy for scientists to study complex processes. Natural selection has favored bacteria that conserve energy by expressing only certain genes and only at specific times when the products are needed. All of our genes don't need to be on all of the time. So how do bacteria selectively turn their genes on and off? So scientists always study model simple organisms and then try to figure out what's going on simply before they study the more complex organisms. They have what's called an operon. Bacterial genomes have clusters of related genes that share a promoter. Promoter is the start site of transcription. And they get transcribed as a single large mRNA molecule. The bacterium E. coli uses the lac operon to coordinate the expression of genes that produce enzymes used to break down lactose in the bacteria's environment. Lactose is the sugar found in milk. So if bacteria encounter lactose, then they turn this lac operon on, this set of genes. If there's no lactose, there's no point in turning this on, so they don't waste their energy. So this is only when lactose is present in their environment. So structure, all operons have the same basic structure. A promoter, like I said, that start sequence, it's the control sequence where the transcription enzyme attaches and initiates transcription. An operator, which is a DNA segment that acts as a switch that gets turned on and off. And then a repressor. To repress means to like stop something or block something. So the repressor binds to the operator and physically blocks the attachment of RNA polymerase, which means transcription cannot happen. So it physically blocks transcription from happening. So you promote something, you want to get it started, that's the start site. The operator is the on-off switch that needs to be turned on to actually operate. And then the repressor, to repress means to block something. You always hear about repressed memories, memories being blocked by your brain. So a repressor actually physically blocks transcription. Then the lac operon in action. We're going to put it all together here.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
Gene regulation in eukaryotic cells like us. So the lac operon is a very, very, very simple model that happens in bacteria. Our cells are a little bit more complicated. So eukaryotic cells have more complex gene regulating mechanisms with many points where the process can be turned on or off. The multiple mechanisms that control gene expression are like the many control valves along a water supply. So a lot of you probably haven't seen the water supply in your house. There's different points where the water can get turned on and off. That's kind of what the eukaryotic genome is like. The initiation of transcription is the most important stage for regulating gene expression. In prokaryotes and eukaryotes, regulatory proteins bind to DNA and then turn the genes on or off. So the repressor, like we said, physically binds to the DNA at a certain point and blocks transcription. Something has to happen to let that repressor, to make that repressor let go so that transcription can happen. Cell signaling. In a multicellular organism, gene regulation can cross cell boundaries, and this is how cells communicate with each other. A cell can produce and secrete chemicals, such as hormones, that affect gene regulation in another cell. So our cells aren't by themselves. Our cells are surrounded by other cells, and they have to communicate to be able to function properly.
Our transcription animation provides an overview of the major steps involved in RNA production. In this animation, we will take a closer look at the events that prepare the DNA for transcription. mRNA and eventually proteins are produced when and where they are needed in an organism. Some genes, such as those for housekeeping proteins in processes like glycolysis, are expressed all of the time. These genes undergo what is known as constitutive transcription. Other proteins are only produced at specific times in certain cells. In these cells, genes undergo a process called regulated transcription. Transcription is regulated by proteins called transcription factors. These factors are proteins that are produced in the cytoplasm and eventually migrate into the nucleus, where they interact with DNA and activate transcription. These transcription factors only interact with specific genes, those genes whose transcription they control. Most eukaryotic species have over 1,000 transcription factors. Regulated transcription begins when a signal is received by the cell. This signal, often a protein, begins a signal transduction cascade that lets the cell know certain proteins are now needed. The most common activation event for a protein in a signal pathway is the addition of a phosphate group. That activated protein in turn interacts with another protein. This interaction leads to the phosphorylation of the next protein in the pathway. There is often a series of proteins that are activated by phosphorylation. At the end of this cascade, the final protein will be activated and then enter the nucleus. It must make this journey because transcription only occurs inside the nucleus. After entering through a nuclear pore, the protein interacts with the specific transcription factor responsible for activating mRNA production. As with the earlier proteins, the transcription factor is modified through phosphorylation. The activated transcription factor will next bind to an enhancer region. The enhancer is a region of DNA upstream of the transcription start site that binds a transcription factor. The transcription factor then binds to the DNA and moves to interact with the rest of the transcription protein complex, located at the transcription start site. When the formation of this complex is complete, transcription of the gene will begin. It is this sequence of events that ensures genes required for a specific tissue or only at a specific time are expressed appropriately.
So you guys should have all of these videos if you need to rewatch them. There we go. So homeotic genes, also called homeobox genes or Hox genes, they're master control genes. They regulate groups of other genes that determine what body parts will develop in which locations. Mutations in homeotic genes can produce bizarre effects. And similar homeotic genes can help direct embryonic development in nearly every eukaryotic organism examined so far.
So what do we learn by looking at 600 million years of animal history? Evolution's tinkering with mammalness to make whales. In the same way, it's tinkering with fishiness to make tetrapods. And it's tinkering with animalness to make all the different body plans that we see. All these different creatures are variations of the same theme, restated over and over again. The question was, what was evolution tinkering with? One of the remarkable discoveries of the last 20 years is that evolution's not tinkering with the bodies. It's tinkering with the recipe, the machinery that builds bodies. What is that recipe? What is that machinery? It's the genes.
Scientists had long suspected that embryos held clues to how animals evolve. All embryos start out as clusters of nearly identical cells. But soon, an embryo partitions itself into specialized segments, which develop into the final form of the animal. What controlled this process? How did the embryos know what shape to take? One researcher, Dr. Ed Lewis of Caltech, studied this question for 30 years by crossbreeding thousands of flies. Lewis's work led him to a controversial idea. He proposed that a surprisingly simple mechanism was shaping embryos. He wrote that each segment of the fly was being directed to grow by a single gene. A small set of genes, a kind of genetic toolkit, appeared to be laying out the entire body. In 1994, Walter Gehring of the University of Basel isolated the gene that triggered the growth of eyes in fruit flies. The gene was called eyeless because flies without it developed with no eyes. Gehring knew of a gene in mice that worked in the same way. He wondered, were the two genes the same?
And this question we tested by taking the mouse gene and putting it into fruit flies to see whether the flies can understand the message of the mouse.
Gehring replaced a fly's gene for eyes with the mouse gene.
And to everybody's surprise, the mouse gene works perfectly well and can induce the compound eye in the fruit fly.
The fruit fly grew normal fruit fly eyes using a gene from a mouse. Not only did the two creatures use the same mechanism, they used the same gene.
So what this means is in some ways, some sense, evolution is a simpler process than we first thought. When you think about all of the diversity of forms out there, we first believed that this would involve all sorts of novel creations, starting from scratch, again and again and again. We now understand that no, that evolution works with packets of information and uses them in new and different ways and new and different combinations without necessarily having to invent anything fundamentally new, but new combinations.
Suddenly, the commonality of form among animals was understood. Animals resembled each other because they all used the same set of genes to build their bodies. A set of genes inherited from a common ancestor that lived long ago.
And there's only one inescapable conclusion you can draw from that, which is if all of these branches have these genes, then you have to go to the base of that, which is the last common ancestor of all animals. And you deduce it must have had these genes. So the whole radiation of animals, the whole spring of animal diversity has been fed by essentially the same set of genes.
So what do we learn by looking at 600?
Oh, next one. So stem cells are undifferentiated cells. They're in very, very early development. They all contain a complete genome and all have the potential to express all of an organism's genes. So stem cells are all the same kind of cells, and all of the genes are switched off. That's only when the genes get switched on when you get the specialized cells. Stem cells have a ton of genetic potential. Scientists use them all the time. So the somatic cells of a single plant can be used to produce hundreds or thousands of identical organisms, which are clones from that original plant. Plant cloning demonstrates that cell differentiation in plants is reversible and does not cause irreversible changes in the DNA. Plant cloning is used extensively in agriculture. So this is our first attempt at cloning was with plants. So cells get removed from an orchid plant, from a little tiny stem. Scientists can grow them in a medium. They perform cell division, they start dividing just like normal. And then a tiny little plant grows. And eventually that tiny little plant grows into the exact same orchid plant that you had in the beginning. Animals can also do this. We can't, but some animals can. It's called regeneration. It's the regrowth of lost body parts, and it occurs, for example, in the regrowth of the legs of salamanders or arms of starfish. Everybody's heard that if you cut off a leg or a tail of some kind of reptile, it'll grow back. Or starfish, if you cut off the arm of a starfish, the starfish will regrow that arm, but the arm will also regrow an entirely new starfish. During regeneration of a limb in certain animals, there are steps, cells in the limb. They reverse their differentiated state to become undifferentiated, or basically stem cells again. They go through multiple cell divisions, and then they differentiate again into the various tissues to give rise to a new limb. So you need bone and tendons and muscle and skin. So they go backwards at first into stem cells, and then they reprogram into all of those different kinds of cells it takes to make a limb. The reproductive cloning of animals. Nuclear transplantation is the process to make this happen. And humans have cloned animals. They have not cloned other humans, but they've cloned animals. So it involves replacing the nucleus of an egg cell with the nucleus from a differentiated cell from an adult body, and then allowing the egg to develop into an adult. So they basically take the nucleus of the egg cell, take it out, and then replace it with a cell that's already differentiated, and then they just let it grow and see what happens. In 1997, Scottish researchers produced Dolly, a sheep, by replacing the nucleus of an egg cell with the nucleus of an adult somatic cell. And that is reproductive cloning, cloning an entire animal. You guys can watch the video on Dolly the sheep if you want later. That was the very first animal that was ever cloned.
headline on the other major.
Since dolly, reproductive cloning has been used to clone many species of mammals, including mice, horses, dogs, mules, cows, pigs, rabbits, ferrets, and cats. Reproductive cloning has been used to restock populations of endangered species, including a wild mouflon, which is a small European sheep. A banteng is a Japanese cow. A gar is an Asian ox. And gray wolves. These are just pictures. If you've never seen a mouflon, a banteng, or a gar, that's what they are. However, cloning does not increase genetic diversity. Clones have the exact same DNA every single time. Genetic diversity is essential to long-term species survival. So unless we're taking care of them, these cloned animals most likely would not survive out in the wild. Genetic variation ensures that in the event of some disaster, fire, flood, disease, some individuals of a species will survive. I don't know if you guys have seen the movie I Am Legend. We usually watch it at some point in the year. But it illustrates that last bullet point perfectly. There's a disaster, a virus sweeps through the human population, kills almost everybody, but there's this tiny, tiny little percent of the population that have some kind of mutation or variation in their blood that makes them immune to becoming a zombie. And that tiny population was just enough to save the human race and not go extinct from not going extinct. Human cloning. The cloning of mammals has heightened speculation about human cloning, but it is very difficult and very inefficient, and there are obvious ethical objections to human cloning. So no, humans have not been cloned yet for various reasons. Therapeutic cloning is what most scientists concentrate on. And the purpose of therapeutic cloning is not to produce a viable organism, but to produce embryonic stem cells. These are derived from blastocysts and can give rise to all the specialized cells in the body.